Hello, in this video we will discuss about the ATP synthase mechanism for synthesis of ATP. So remember, this is the inner mitochondrial matrix contain less proton when the oxidation through electron transport chain, while the outer mitochondrial matrix when proton will increase will lead to decrease the pH, mean more acidic, while the inside is the more basic. So here is the ATP synthase contain inner matrix domain and the the membranous domain, the mitochondrial membranous domain. So F1 and F0 we can divide. F1 contain alpha and beta subunit respectively and used for the ATP synthesis, while the middle is the gamma and epsilon, while this is the subunit is the C10, while the proton is pumped from outside to the inside through a alpha subunit. So it contains arginine 210 and the gas 961. So here you can see that when proton passes make gamma spin within alpha and beta head. So within alpha and beta head, the gamma will spin used for the synthesis of ATP. So how it is possible? Let's begin to understand. So this is the rotation of the gamma subunit, which that is attached with the epsilon. So remember, when in spin will actually produce the ATP from the ADP, we will also learn about the mechanism with the details. So here I am just recapping the binding change, the mechanism, ATP synthesis we will understand now. The mechanism, so the binding change mechanism when bind the ADP with inorganic phosphate. So here you can see this is the six subunit of the alpha and beta respectively inside, and here is the OTL site, and inside is the gamma used for the spinning. So the alpha, beta and alpha sub respectively repeat. So here you can see this is the cross section of the F1 particle, which that is I am describing, and basically is the delta is also involved for the F1 particle containing, here you can see, and B2, which that is showing in the diagram. I am not describing about that, but we just understanding about the binding change mechanism and ATP synthesis. So first is the O mean open, and the open mean number one, we will discuss the open side. The new ATP is released after the formation of ATP, and the ADP and the inorganic phosphate bind. So the inorganic phosphate will be bind, remember, and the ADP in the beta subunit of the F1 domain. And this is the O site, remember. The O site is very important for the binding new ATP is released, and the ADP and inorganic phosphate will be bind. So remember, after this the inorganic phosphate will be bind with the ADP. Basically it is not bind, it is bind with a beta subunit. So after this it is here is a loose state. This is the step 2, which that is a bound ADP and inorganic phosphate cannot be released because it is attached. So remember, it cannot be released in the loose side, in the loose stage, step 2. So after this step 2, the step 3 is involved. This is the tight, and the tight means the condensation of ADP, and the inorganic phosphate is favored to form ATP. The ATP, basically it is favored to form ATP from the ADP. The ATP formed is very tightly bound, and in this way the formation of ATP, which that will be released in the open site when the new ATP is released, remember. So O side will release and additionally again add the ATP, so the continuously reaction will occur. So the tight means the condensation of ADP and inorganic phosphate is favored to form ATP. So the ATP is favored to form ATP. The ATP form is very tightly bound and released into the inner mitochondrial matrix. So when released into the inner mitochondrial matrix, it is move out from the mitochondria because it is basically a smaller molecule can pass from the inner mitochondrial matrix to the cytoplasm. So it is used for the synthesis, is used for the production of heat, it is used for the synthesis and the maintenance homeostasis and other things, as well as ATP is never possible to store, so it is continuously working. So anyhow, here is a beta subunit, which that is the T side is the tight formation, will form the ATP. And this ATP will not release still as long as when it will go into the open side, the open, when it will be open. So the open to add more ADP, and this ATP will release to add more ADP. So this new ATP is released and ADP and inorganic phosphate bind again. So in this the O side, basically the O mean the open and L mean the loose. The loose formation will lead to T is a tight formation. So the tight formation. So the rotation of this inside, the rotation of this inside, the gamma subunit in the F1 particle is actually rotate, while the outer is the basically the inside is the rotate, but the remember that is rotation will cause